Thank you, Elijah, for that wonderful talk from Dudutech. As Victor had mentioned earlier on, I want to mention that I have a number of colleagues joining in also in this webinar. One of the panelists is Bana Barotich. So we are doing this together with the Syngenta group. So mine would be to briefly talk about two key areas. I'd like to highlight thrips biology, linking with what Elijah said and with the implications on management mainly. And secondly, I would like to look at what are the biological control options that we have at our disposal for the management of thrips. Next, please, Liz. So this is an overview of my presentation for today, which should take around 15 minutes maximum. So we want to look at thrips, what they are, look at the biology. Then I'll look at what are the management options at our disposal, cultural, physical, and then I'll end it by looking at the biological options as well, under which we want to look at predators, entomopathogenic nematodes, and fungi. The world over, these are the three groups of biocontrol agents that are used in thrips management. So we'll be highlighting that at the end. Next, please, Liz. So if you could dive straight on to thrips, what are they? So thrips belong to the insect group called Thysanoptera. It's an order, really. So when you are classifying insects, the order are based on the wing morphology or the wing structure. So that name Thysanoptera simply means that the wings of thrips are like eyelashes. They have a stalk and then the hairs grow out of it. Please, if you can play the video on the right, on the bottom right, there's an adult thrips and you'll be able to, the viewers will be able to see what I'm talking about, the wings of thrips. Then thrips are the smallest winged insects. Unfortunately, small but causing a lot of damage. And a bit of semantics, a bit of language issues. There's no such thing as thrip. Many times I talk to growers and they say, you know, thrip is a problem. There's nothing like thrip. Correct English should be thrips, whether it's one or whether there are many. More seriously, we have more than 6,000 species of thrips worldwide. It's such a diverse group, including some that are even used in biological control, as I'll mention later on. But out of the 6,000 species, 100 species have been recorded to be pests of crops that we are growing, out of which the damage cost is estimated to cost $10 billion worldwide on an annual basis. So we are talking about a small insect that is really destructive. Next, please, Liz. So talking about pest thrips, which is our focus for today. Most of the pest thrips belong to this family called Thripidae. Again, Thripidae is a diverse group, including some that are feeding on spider mites and not feeding on crops, as you can see the third last point. But I'd like to draw our attention to two main genera in this group, Thripidae, and that is Thrips and Franklinella, the second last point. So straight away, when you talk about thrips, there's a genus called thrips, like Thrips tabaci, the onion thrips. Or Franklinella, we have here of Franklinella occidentalis, what we love to call Western flower thrips. And we also love to hate it the most on our flowers. And talking about the Western flower thrips, you can briefly highlight on that. It's called the Western flower thrips because it's an import from the western side of America, the western coast. Hence the name Western flower thrips. It originated from the western coast of the USA. Currently, thrips experts are warning us that there's an eastern thrips, again, eastern flower thrips, coming from the eastern side of the USA again that is spreading worldwide, Franklinella atricity. So there's a warning that that is also spreading all over the world. Next, please, Liz. So how is the life cycle of thrips? Elijah has mentioned something on this. I would just like to highlight issues that I think have implications for management. So like Elijah mentioned, thrips has 6 developmental stages ranging from egg up to adults. The eggs are laid underneath the epidermis. It's actually inserted into the tissues of the plant. Thrips has a soul-like ovipositor. Ovipositor is that organ that they use to lay eggs. So it actually lays its egg inside the plant. Take note, inside. So Elijah mentioned something about cryptic. So the hiding starts from the eggs. The first instar nymphs that hatches from the egg loves to live inside the very tight spots. If you remember the word thigmotactic behavior that Elijah mentioned earlier on. So they like hiding in the very tight spots inside the flower buds. Or if you have leaves, those unopened leaves, that's where they like to hide. The same case with the second instar nymph. It molts the second instar nymph, still hiding inside the flower petals. The prepupa is the next stage. This stage drops to the ground. So pupation takes place up to 5 millimeters below the soil surface or below the media surface, whichever media you're using. So that's where you find the pupa and the prepupa. So if you're following me, you'll realize that all those stages, there's no stage that is easily accessible. And that has serious implications for management of thrips. The adults is what comes out of the soil or the media for that matter. And they love to stay inside the flower buds again. Elijah mentioned that they are diurnal, they come out during the day. So what comes out really most of the time are the adult stages. The nymphs would spend most of their time inside the flower buds. To worsen the case for management, thrips also have a very rapid life cycle. Egg to egg can take between 1 week to 2 weeks, 7 to 13 days. And if you're talking about resistance development that Daisy is going to talk about later on, we find that resistance develops very fast in insects that have such a short generation time. Next, please. So just still highlighting on the life cycle, remember I've said virtually all these stages, there's none that is easily accessible, and that's why management is a bit of a challenge. So there are stages that you'll find in the soil, there are stages on the plant, I think I've highlighted that. So the pictures on the right indicating the different stages. So the very small one at the top is the first instar, the second instar, and the adults. Those are the stages that you'll find on the plant, represented here by the green part of the pie chart. Then the prepupa and the pupa. The one on the left is the prepupa, when the wing buds have started developing. Then you have the pupa when the wing pads have already elongated. Next, please. So if you are to talk about the generation time for thrips, this is a very ideal scenario that is not likely to happen in nature, but it serves to highlight what we are talking about, the exponential capability of thrips to develop. So if you're starting with 1 thrips eggs, assuming it's a female, given the biology that we just talked about, within 12 weeks, we could be ending up with 85.75 trillion eggs if every condition is perfect, which of course nature is not perfect, but it just serves to highlight the fact that thrips are able to develop very fast within the greenhouse if the conditions are right. Next, please. So what damage do thrips cause? The damage that is caused by thrips comes about due to their feeding behavior. So if they're feeding on the petals, there'll be direct damage. If they're feeding on the petals or the leaves, wherever they are, there'll be direct damage and indirect damage. So talking of direct damage, thrips have piercing and sucking mouthparts. Again, for a long time, literature has told us that, thrips has rasping mouthparts. They rasp and they suck the sap. It's not very true. They actually have piercing and sucking mouthparts. They pierce the individual cells and suck the contents of the cells. So what you see as a discoloration of the petals or the leaves is as a result of the dead cells that have been filled up with air, and that's what appears as white or off color on the petals that we observe. Next, please. So still on direct damage, it will be something similar for the leaves again, or the green parts of the plant. If you look at that leaf on the top, I think that's chrysanthemums. It looks quite like mite damage. The difference between thrips, the critical difference between thrips and mite damage, thrips typically would start from the margins of the leaves going inwards, while mites will always start around the midribs or the midvent for that matter. Next, please. So in terms of indirect damage of thrips, the vector, a genus of viruses that we call tospoviruses. There are a number of them, but the key most important ones are the two that are highlighted, tomato spotted wilt virus, and you also have Impatiens necrotic spot virus. So this is the group that we call tospoviruses. For a long time, we've always believed that these viruses only affect solanaceous plant. From my reading, it's recorded that these viruses have been recorded in more than 80 families, not just Solanaceae. And they affect more than 800 species. So even for us rose growers, we need to watch out. Maybe very soon we could find this virus on our crops as well. Then apart from the virus vectoring, they also passive vectors, passive carriers of fungal infections. I think if you talk to any grower, they'll always associate thrips with botrytis. And for a good reason, thrips cause those injuries on the petals that act as entry point for the fungal infections, as well as bacterial infections or secondary infections for that matter. Then also to mention, some thrips are notifiable pests. A good example here is another thrips genus called Thrips palmi, which is not acceptable in the EU, where most of us are exporting our floricultural produce. Next, please. So as we come to the end of this, how do we manage thrips then? I want to start by an admission that thrips management is challenging for a variety of reasons that have been highlighted by myself and earlier on Elijah. But like Elijah, I would have to go with him and say that we have not really reached an impasse. I'm an optimist, just like we trounced red spider mites. I believe if we do the right things, we can trounce thrips as well. So I'm an optimist. As an optimist, I go by the saying by one of Africa's greats. The next slide, please. Nelson Mandela. I always remember this quote. After climbing one great hill in life, we only find that there are many more hills to climb. So life is continuous challenges. And I believe without challenges, life would not be interesting. So in growing, likewise, we don't expect easy solutions. We climb hills and new hills come up, thrips, FCM and the likes, and we have to keep climbing. The question is, do we have the necessary arsenal to keep climbing? And that's why we are coming together to present such fora in an attempt to develop and come up with solutions. Next, please. So the way out of thrips management, just like with red spider mites, we can't rely on these solutions, as Kenyan growers we like to say. I think I've attended many seminars and growers would always ask, so what is the solution of this problem? We don't have these solutions, but rather we have a combination of solutions that should be used together with the right knowledge to come up with a solution. So in terms of thrips cultural management, sanitation ranks high. Like with any other pest, it's always recommended that you start clean planting material, clean growing media, clean greenhouses. That's the starting point. And once we've started clean, we have to make sure that we stay clean. So where feasible, we could screen out the thrips by using netting material that is very fine that thrips are not able to go through. But for any grower, you'd realize that when you do that, the very fine netting would also compromise ventilation. So we need to find a way of optimizing ventilation within the greenhouse if you're going to go the screening way. Weeding, thrips are polyphagous. To use Elijah's word, they feed on a variety of plants, not just the flowers. So make sure that you don't have anything that is flowering in and around the greenhouse. Then mulching is also another method, particularly covering the media so that the thrips do not get access to the pupation site. Then there's been some interesting research also on mulching where we use UV repelling mulches just to confuse the thrips so that they do not land on the crop. That's an interesting research that is also coming up. Removal of debris and open flowers, like you can see on the right. So again, for the debris, we do so well in removing them from the greenhouse, but most growers have been to, they place them outside of the greenhouse in the open. So again, thrips will realize that the thing is drying up and they'll fly back to the greenhouse. So it's always recommended that you keep them covered, either wrap them in a polythene bag or have a drum that is you can close to keep them off from getting back. Then there's a controversial one on the bottom right. Avoid wearing blue or yellow clothing in the fields. We all know that yellow and blue attract flying insects. Blue particularly for thrips. So if you're doing this, what these guys are doing, then we could end up spreading the problem even where it's not there. Then I also wanted to mention something on repelling in the times of staying clean, the use of repellents. And there's a very good product that is gaining a lot of prominence also, garlic extracts from Dudutech. It's called Nemgard. We have it for nematodes, but for some reason, guys realize that it repels thrips very well. So when it's combined with pesticides, either through an application before the pesticide application or as a cocktail, then we tend to have more lethal effect on the thrips. It kills more. When you're combining with sticky cards that I'm going to talk about next, you end up catching more because thrips are agitated to come out from their hiding places. Next, please. Then next, I'd like to talk about mass trapping. Like you can see on the right, most of us are doing yellow or blue sticky traps that works against thrips. For those of us who have been doing that, I have good results from a peer-reviewed publication that actually indicates that traps can go a long way in thrips management. So if you look at the graphs from this peer-reviewed paper from Professor Kirk and Sampson, the graph on the left is looking at the population of thrips that are found in the flower. And you can see 4 is the damage threshold, is the threshold that they're looking to keep the thrip population below. So what this shows us is that simply by the use of traps alone, whether traps only relying on color or traps that are combined with aggregation pheromones, these guys were able to keep thrips below the damage thresholds. This applies to fruits, specifically strawberries. So if you can do this in strawberries, straight away you can see that trapping would have a huge impact in thrips management, even in flowers. Then the one on the right is now gauging the damage that is caused by the pests. It would imply that if you have fewer numbers of thrips per flower as a result of trapping, then you're also going to end up with less damage, as you can see with the graph on the right again. I read this paper and I didn't include another graph that actually shows that just by mass trapping, we were able to end up with more sellable fruits. So again, economic benefits derived directly from the use of traps alone. Next, please. Also very important in thrips management is is monitoring. So 2 methods of monitoring. We can use color traps, yellow or blue. There was a question from the morning session. Blue tends to work very well for the clear polythene. That's current research that is also coming up. While yellow tends to work well against thrips for UV blocked polythene. So mostly what we use in the tropics, I think what is coming out is that yellow tends to trap more. Again, the best way is to try it out yourself and see what works best. But this is the research that is coming out recently. Then still on cultural control, environmental control is yet another method. Thrips don't like it when the humidity is high or when the growing media is very wet. They don't like it. So overhead irrigation through showering, misting would also go a long way in thrips management. Next, please. Good, so that covers physical and biological, physical and cultural controls. So as I come to the end of my presentation, I'd also want to talk about biological control. This would be an important cog in thrips management. And actually, the whole idea of Syngenta and Dudutech coming together is the realization that we don't have silver bullets in thrips management. We need to combine different methods. We can't succeed with biological control alone, as much as I'm coming from Dudutech. We can't succeed with chemical control, and that's why we are joining up with Syngenta. Neither can you succeed with cultural or physical. We need a combination of methods. So biological control would be an important weapon also in your thrips management strategy. It will also help in overcoming some of the challenges that Elijah highlighted earlier on. Key one being resistance. You'll never hear of a pest developing resistance to biological control agents. Apart from the obvious effects also, biological controls are safe to the environment. So it's a very important method also in the management of thrips. So I'd like to start off with predators. If you remember in biological control, I mentioned 3 methods. We can use predators, we can use pathogens in the form of nematodes, or we can use pathogens in the form of fungi, what we call entomopathogenic nematodes or entomopathogenic fungi. So I would start off with a new kid in the block as far as predatory mites are concerned. And this new kid in the block, it's called Transeus montidorensis. Initially, it used to be called Impatiens montidorensis. So again, some scientists believe that taxonomists are some of the most idle group of scientists. So when they get tired of the name Impatiens, they change it to Transeus. We have to live with it, unfortunately, they're the experts. So the name has changed to Transeus montidorensis. So Transeus montidorensis was actually first discovered in Australia. It's a mite, a predatory mite, as I've already mentioned, that feeds on the young thrips, the nymphal stages that I mentioned earlier on, the first and the second insta nymphs that are found within the flowers. It's a very tiny mite, so it's able to get the nymphs from their hiding places deep inside the flower buds. It feeds on a range of pests, so you're not only controlling thrips, you're also able to control mites, you're also able to control whiteflies. And when I talk about mites, I'm not just talking about red spider mites. I'm talking about a group of mites, including the rosette mites and the other tiny mites that you find on the crop. They're able to survive even when the prey is in low densities. They're not only relying on thrips and these other insects, they're also able to feed on pollen. They prefer warm conditions, but they're able to survive both in cool and warm conditions. As with any other biological control, we recommend that they are introduced earlier in the cycle so that they establish before they are overrun by pests. They work well preventatively as opposed to as a firefighting tool. Next, please. Still on predatory mites that are used on the foliage stages, we also have Impatiens cucumeris. Again, the name has changed. It's going to be called going forward Neosalus cucumeris. So it also feeds on the young nymphs, particularly the first insta nymph. Best used before the pest establishes. It prefers cooler conditions. And if you have a crop that will have a source of pollen, the better for you. It establishes very fast when you have a crop that is rich in pollen. Next, please. The last one I'd like to talk about under these foliage predators is Impatiens sweski. Again, it's another Impatiens. It feeds on thrips nymphs. It feeds on whiteflies as well, so you're not only controlling thrips. It's likewise also able to survive on low prey densities. And I dare say this group of predators are also fairly resistant to most pesticides. They're not like your phytophthora, so they're quite hardy. They're able to survive when it's hot and also when it's cooled, and they're also able to survive a number of pesticides. Again, it is best for crops that have a lot of pollen, capsicum and the like. Next, please. So this particular slide is very important. We are now comparing these foliar predatory mites. How do they compare in terms of life history parameters? As I mentioned earlier on, this exciting mite called Transeus montidorensis seems to be towering head and shoulders above the rest. So it's generating a lot of interest for a good reason. And I want to mention, just for your information purposes, as Dudutech, we do not want to be left behind. So we are actually in the process of developing Transeus montidorensis for you guys, for the market against thrips. So as you can see, and these are peer-reviewed papers actually, you can refer, I think the presentation will be shared earlier on. If you're interested, you can read these papers. You could realize that Transeus montidorensis trounces all these other predatory mites in almost every parameter, whether you're talking about the predation rate, the oviposition rate, either on pollen or on thrips, how the intrinsic rate of increase, how fast are they able to multiply on the crop. We can see that it's performing really well. Next, please. So that is on the life history parameters. But how about in doing the real job? And the real job, of course, is managing thrips. How does it compare with the rest? Again, these are peer-reviewed papers. There's actually a reference. This is a paper that was published last year, 2019, by Dr. Labe. I think the reference would be able to show in the presentation that will be shared later on. So if you look at the graph on the left, again, if you look at the number of thrips that are found per plant, and you're comparing these 4 different Amblychias mites, or predatory mites for that matter, again, you can see that the graph below indicated TM, Transeus montidorensis. Transeus montidorensis is able to maintain thrips population very low on the plant compared to the controls and compared to the other mites. Are we able to find the mites on the plant after application? So in your scouting reports, your scouts should also be able to report whether they're finding natural enemies or not. So this is what the graphs on the right is showing. Again, you can see that Transeus montidorensis is coming out top. It's actually very visible, very much visible on the crop. And it's not just a question of visibility, it's also doing the good job as it's indicated on the graph on the left. Next, please. So still on predators, again, I'll take you back to the life cycle of thrips. Remember we said that the pupal and the prepupal stages are found in the media, in the soil. So how do we take care of this critical stage as well? Victor will tell you that if you have to use a chemical, it may not make economic sense. So you'll rather have something that takes care of it and is able to multiply naturally. And that's where we have Hypoaspis miles. Again, the name is changing. It's going to be a mouthful. Stratiolaelaps scimitars, that is the new name. So again, we are sticking with the old name for now. So our brand is called Hypotech, Hypoaspis miles. But the correct name scientifically now is Stratiolaelaps scimitars. It's a soil-dwelling predator, as I've already mentioned. So it's actually applied to the media and is able to establish very well on a variety of growing media, whether it's the soil, pumice, coir, or coconut coir, and the likes. Again, it's best introduced earlier in the crop cycle. It doesn't like dry soils or dry media, neither does it like waterlogged media. So the humidity should just be right. It should not be too wet. It should not be too dry. And then you have the extra benefit of also taking care of the nuisance pests, the sclarid flies, the fungus gnats, the sawflies. And for those of us who are doing chrysanthemums, it's also able to take care of your leaf miner, the pupal stage that is found in the soil, and also the stages on the leaves. Not on the leaves really, but in the soil, sorry. Next, please. So that wraps up our predators. So as I come to a conclusion, I'd also like to talk about the pathogens, and starting off with entomopathogenic nematodes. That big word simply means insect feeding or insect killing nematodes, nematodes that are able to kill insects. If you can look at the cycle on the right, that's how nematodes work. So here we have a product called Nematec S, S for Stenanema feltii, as indicated here. And that's basically the life cycle. It entered into the insects. It has a bacteria that it uses to kill the insects, and then it feeds on the bacteria that has multiplied within the insects. So straightforward life cycle. Again, used correctly, we've been able to get very good results simply by the use of Stenanema. And the goodness, Stenanema is what you use and it stays working for more than a month. So we've been able to record very good efficacy, not just on the pupal stages in the soil, but also on the foliar stages, the nymphs and the adults. If you do high volume misting or sprenching, as we like to call it here, spray that gets down to the media. So again, we need very high humidities. So our recommendation always, like with any other biological control, preferably apply late in the evening when the humidity is high and when the temperature is just right. Next, please. Finally, we also have fungi that can be used to control insects. We call them entomopathogenic fungi. A good example here is Beauveria bassiana. Our brand is Buvitech. We also have Lecanicilium lecani, Lecatech. So this group of fungi are generally broad spectrum. They'll target a wide variety of insects. So as you can see, the picture shown there is Beauveria attacking bugs, or what you call heteropterans, causing what you call the white muscadine disease. At the end of the day, the insect dies and you have that white growth, fluffy growth growing from it. Best effective at very high humidities. Again, we recommend humidities of above 70%. Again, evening application would make more sense. Next, please. And that brings me to the end of my presentation. The key takeaway that I'd like us to go home with, there are no silver bullets. We have to do the work in order to come up with a solution that works. And the solution that works is not single solutions, it's about combining different solutions. It's not about the solution, but it's about solutions in combination. And that's what IPM is all about. Thank you very much.